Welcome to our demonstration of urine drug screen testing. This is a preliminary screening test that detects drug of abuse in urine at specified detection levels. Many research studies sponsored by the National Institute on Drug Abuse Clinical Trial Network, or NIDA-CTN, use a urine drug screen test as a usual assessment. It frequently is a primary or secondary outcome variable for the clinical trial, so it is an important assessment. Therefore, it is critical that you learn how to do the assessment precisely. The urine dip card used for the CTN studies tests for the same drugs of abuse across all CTN research trials, so that comparisons may be made between trials. Prior to a visit, check to see whether a urine sample will need to be collected by verifying in the schedule of assessments what tests are to be performed at that visit. If urine collection is required, ensure that you have all necessary supplies prior to the visit. The QuickTox drug screen dip card tests should be stored at room temperature, 60 to 85 degrees Fahrenheit. Verify that the supplies have been stored appropriately, no temperature excursions outside of range, and the test dip cards have not expired. Notifications of expiring materials are sent by Eminent, the central distributor for the Clinical Coordinating Center, CCC, at EMIS. However, if expired materials are found, try to find other supplies to use that have not expired and separate the expired materials from the study supplies. You may contact the assigned protocol specialist at EMIS for instructions regarding destruction of expired supplies and to inquire about replacements. Let's talk about the supplies needed for the urine drug screen testing. As you see, we have a urine cup with an attached temperature strip, an adulterant test strip, a laminated color chart for interpreting the results, and the 10-panel urine drug screen dip card. Other supplemental materials that may be needed for performing the test include gloves, paper towels or absorbent sheets, timer or stopwatch, disinfectant for cleaning the area. So, let's get started. Your participant is here. Hand them a urine cup and instruct them to fill it with urine at least half full or more. If a participant is not able to provide an adequate sample now, you can give them a glass or two of water and let them wait until they feel ready to provide a urine sample. Now, let's proceed with doing the actual test. This is a 6-step process, and we will explain and demonstrate each step. Once an adequate urine sample has been obtained and you have put on your gloves, check to make sure that the temperature on the strip is within the adequate range of 90 to 100 degrees Fahrenheit. Temperature is important, and a reading that is above or below the expected temperature range may indicate that the participant has substituted the sample with urine that did not originate from his or her own body. Any sample that is outside of this range is considered adulterated. If the temperature strip shows a reading within range, 90 to 100 degrees Fahrenheit, conduct the adulterant test using the Urine Check 7 adulterant strip. Remove a strip from the Urine Check 7 cylinder and replace the cap. Immerse the test strip completely into the urine and then immediately remove the strip from the urine, tapping once or twice on the cup to remove any excess urine. The adulterant strip tests for different substances, each showing results within a different time frame. Look at the laminated color card provided by the CCC for specific time frames for each tested item. The adulterant test strip will test for the following: creatinine, nitrite, glutaraldehyde, pH, specific gravity, bleach, pyridinium chlorochromate. Read each test at the specified time after dipping. Immediate for pH, 30 seconds for bleach, pyridinium chlorochromate, 45 seconds for creatinine, nitrite, glutaraldehyde, specific gravity. You may need to practice several times until you're comfortable with the timing and the process. Each substance will fall into one of 3 categories: normal, abnormal low, or abnormal high. To obtain results, compare the color on the laminated card to the color seen on the strip and find the best match. If any substance on the adulterant strip is abnormal, that sample is to be considered adulterated. Record this result on the urine drug screen worksheet or other study-specific document. Adulterated samples are not considered valid and should not be used any further. Ask the participant to provide another sample. If they are not able to, you can offer them a glass or two of water and wait until they are able to provide another sample. You may need to counsel them on the importance of giving valid samples. Remind participants that they will not be penalized for providing samples that test positive for substances. Repeat the process until a valid urine sample is provided. Study staff may be required to supervise specimen collection in circumstances where validity of urine samples is in question. If multiple adulterated samples are provided during a screening phase, you may want to consult with a principal investigator about whether that participant is appropriate for this study. After confirming that the urine temperature is in range and the sample has not been adulterated, proceed with the urine drug screen test by removing the 10-panel Quick Tox dip card from its package. Detach the bottom cover by pulling gently and dip the test card into the urine up to the tips of the blue arrows and hold for at least 10 seconds. Do not insert the test card into the urine beyond the arrows. Remove the dip card from the urine, reattach the bottom cover, and lay the device on a flat surface. Results can be read from the device within 5 minutes. Once the control band, C, appears in 5 minutes or less, results are ready to interpret. Look at each test strip separately. Read the results on one side, then turn the device over and read results on the other side. The 10-panel Quick Tox dip card tests for these substances: barbiturates, benzodiazepines, amphetamines, marijuana or THC, methamphetamine, opiates at 2,000 nanograms per milliliter, cocaine, ecstasy or MDMA, oxycodone, methadone. Let's go over the Quick Tox card and show how to interpret the results. This is the control region. Here is the test region, substance names, drug test strip, blue maximum dip level arrows, sample pad, and the bottom cover. And now what you have been waiting for, finally the interpretation of results. For each drug test, 2 colored bands should be observed in the result window. One band at the control region, C, and a band at the specific test region, T. The color of the test band may be slightly darker or lighter than the control band. Any band that can be seen visually, no matter how faint, is a negative result. Read each test independently. Do not compare color intensity of one strip to another. When the control band is visible in the control region, C, and no visible band in the test region, T, the result is presumed positive for that particular drug. When no band appears in the control region, the test is invalid regardless of the test results. There must be a control band in the control region. If the test is invalid, check testing procedures and samples. Repeat the test using a new device. If site staff is unable to read the test results due to a faulty dip card, a new dip card should be used to perform the test again, preferably from a separate lot number. This should be documented in the notes, but only results from a readable test should be collected and recorded. Here are a couple of suggestions based on 2 of the most commonly occurring errors when performing a urine drug test. Don't dip beyond the tip of the arrows, and don't put in urine for longer than 15 seconds. Now, let's think for a minute about a few what-ifs. What if the participant reports no drug use, but the test shows a positive result? This can happen from time to time for several reasons. Check again to make sure the control line for the substance in question is visible. If visible, use your discretion. You may need to test the same urine sample again using a new dip card, preferably from another lot number. If the results change, the original dip card may have been faulty. A third test may be needed to verify the result. If the results do not change from one test to another, it is possible that either the participant is not telling the truth or the result is a false positive. False positives are rare, but they do happen. Check with the PI or a study clinician to see if a medication a participant takes, or something the participant ate, may be causing cross-reactivity with the test. Make sure any discrepancy is well documented. What if multiple tests show as false positives or multiple tests come back invalid? Again, this is very rare, but if you notice a trend with false positives or invalid tests, immediately contact the assigned protocol specialist at the CCC and provide to them the lot numbers of the tests in question. The tests may have been stored incorrectly, or there may have been an issue with the tests themselves. The protocol specialist will consult with other members of the lead team and the supply vendor and give you instructions for how to proceed. You may be asked to refrigerate the samples for possible testing at a central location. Mark the urine sample cup with a participant's ID number prior to refrigerating it, and take a photo of the drug test results. The protocol specialist can also help you to replace faulty dip cards if needed. That's it! You are now prepared to conduct a urine drug screening test. Thanks for watching.